Project title: Preparation and characterization of activated carbon using morinda tinctoria stem for textile industry effluent treatment. Team members: Dinesh Kumar A, Magavishnu R, Venkateswaran K. Project guide: Dr. N. Vasiraja M.Tech PhD, Associate Professor, MEPCO Shillong Engineering College. Content: Introduction, literature review, problem statement, objective, project methodology, experiments, absorption study, CCD, regeneration. Introduction: Environment pollution increases with the increasing industrial developments. The textile industry play a vital industry to pollute the water pollution around the world. Major pollutants are dyes and heavy metals. Problem statement: Methylene blue is a safe drug when used in doses less than 2 mg per kg, but it has a potential to lead fatal at doses greater than 5 mg per kg and rarely can cause severe anaphylactic shock. Morinda tinctoria is an evergreen shrub, is one of the most abundant fast-growing plant in many rural areas. So, Morinda tinctoria is selected for using for preparing activated carbon. These are the literature review. There is rice case with 5:1 ratio. It, it yields bed surface area with high bed surface area 2696. Objective: to prepare the low cost and eco-friendly adsorbents to prepare the KOH activated morinda tinctoria stem carbon by carbonization process to identify the optimum level of KMTSC at which the removal of methylene blue dye is maximum, to fabricate the filter prototype which can purify water by using activated carbon in it. This is a methodology. The project process by collection of wood cut into pieces, then poured in crucible, hot air oven, then muffled furnace, heating, and hot air and carbonization done, and mixed with KOH pellets and impregnation, then activation at 440 degrees Celsius, then dried at hot air oven, and activated carbon produced, and methylene blue dye solution preparation, and activated carbon mixed with methylene blue solution and placed it on magnetic stirrer, then placed in centrifuge for separation of activated carbon, and sample collected. Then the collected sample will test in UV spectrometer scan and the results will plot. Materials purchase: methylene blue dye, KOH sample tube and beaker. Process: collection of wood. The morinda tinctoria plant will be collected from local area, then it will cut into pieces into small pieces, then it is dried in hot air oven 110 degrees Celsius and for 2 hours. Then the carbonization, the wood pieces will place it in muffled furnace and set 440 degrees Celsius. Then it will sieve 250 microns uniformly and then mix it with the KOH. After mix it with the KOH, the KOH will activate the carbon at 440 degrees Celsius. This is a product output, and this is a filter prototype model and filter prototype assembly and experiments conducted for physical characterization, FESM, EDX, FTIR and physiochemical characterization. This is the FESM image and without activation and with activation. Without activation there is a small holes. After activation there is many holes, many pore size, and this is the EDX results. After seeing a study, the activated carbon has the high carbon content 68.25%. This is FTR graph and there is 20 carbon bonding. Analyze of physicochemical characteristics. Yield 35% for normal carbon and 65% for activated carbon. This is the number of variables how selected for absorption. Absorption study: 100 ml of known concentration was methylene blue solution. 50 mg absorbents were mixed within beaker and placed in magnet stirrer, and the stirrer will Stir with a constant speed of 500 to 600 rpm with room temperature 30 plus or minus 15 degrees Celsius. After determined time intervals, then the sample will collected and it will put in centrifuge about 6000 per minute rpm. This is the collected sample results. For 75 mg, it reaches the maximum efficiency. This is a without activated carbon results. And after sample collected, the sample will place in UV spectrometer about to scan 700 to 400 nanometer. The maximum wavelength of methylene blue dye is 662 nanometer. This is the curve plotted. These are the results in UV spectrometer without activation and with activation. Absorption efficiency find by C initial minus C final by C initial into 100. After using this formula, the results will plot. That is with activation maximum efficiency of 96.7 at 75 mg per 100 ml. Without activation only reaches the maximum at 31.4%. These are the curve plots. Then CCD experiment conducted. The operating variable for -101, the table shows the selected variables. The formula used for finding the predicted efficiency. Then the optimized value will find 46.778 dosage, 29.5 minute time, and 43.035 degrees Celsius with the maximum efficiency of 99.872. The 3D surface plot shows the dosage versus temperature plot. Red color indicates the maximum efficiency. Time versus temperature plot and dosage versus temperature plot. This is the CCD predicted versus actual variance. Regeneration study. After producing activated carbon, the the same carbon will use a number of times. So first cycle give us output 96.8%. After 4 cycles, then it decreases to 63.6% at fifth cycle. These are the references. Thank you.